Hello, and welcome to Tierah Ruins Things with Science. I am Tierah, and I'm here again ruining another one of your favorite TV shows that mentions infectious diseases by comparing it with the actual science. I have a degree in immunology and a master's in medical science, and I always loved watching movie and TV shows, so I combined them into this podcast. Today, as part of Season 2, that's all about world and civilization-altering diseases, I'm going to feature the 1918 influenza pandemic as portrayed in Downton Abbey Season 2. It's one of the few TV shows or movies that I've seen that tackles the 1918 pandemic itself rather than just some nameless other influenza. I have not watched every World War I film out there, but I imagine there are probably some others that deal with the pandemic, so some of you might be screaming at your phones right now. But this episode of Downton is something that I have seen, and it deals directly with the flu as a central plot point, so I'm going to do it. Also, I refuse to watch or read Twilight, and apparently that's the reason why Edward was turned into a vampire originally, because he was dying of the flu. I don't know, and I'm content never finding out. This pandemic is the boogeyman, though. This is Baba Yaga up there on par with the Black Death in terms of the global fear it caused, with its rapid spread, multiple waves, revisitations, and the scale of death that happened in the span of just a few short years. Downton Abbey is available on CBC Gem, Netflix, Amazon Prime, BritBox, pretty much everywhere you can rent or stream shows and movies. So I'm going to tell you all about how they did.
Hope they're not coming down with anything. The stories of this Spanish flu are too awful.
The 1918 influenza pandemic is frequently referred to as the Spanish flu, but I will never call it that except to ruin that name and point out that it is wrong. Sometimes names just stick due to publicity or who spoke about something first, whether they're accurate or not. And that's what happened here. It was not Spanish at all, and you're never going to believe where it actually started. The first outbreaks of a flu-like illness were reported in March 1918 at Camp Funston in Fort Riley, Kansas, in the United States. More than 100 soldiers were sick, and within a week that number was over 500. The U.S. had only entered the First World War in April 1917, and by May 1918, a year later, hundreds of thousands of soldiers were crossing the Atlantic every month, and that is how it spread so fast. There were two initial waves, a milder one in the spring, and then the truly deadly autumn wave that began in September 1918. In October 1918, the influenza killed almost 200,000 people in the U.S. in just that month, and by this time it was hitting Europe as well. Eventually, through all the waves of the pandemic, estimates of total mortality are anywhere from 50 to 100 million deaths. The French called it a purulent bronchitis, or the American flu. The Italians called it sandfly fever, which makes me kind of feel like I just said something racist. And the Germans called it the Blitzkatar, which is hilarious and would make a great band name, or the Flanders fever. It's kind of a thing where a lot of bad diseases that spread around get called as being from one country or another. Syphilis was big for that. It was called the French disease by the English and the Germans. The Polish called it the Russian disease. The Dutch and the Portuguese called it the Spanish disease, etc. As this flu broke out, both Ally and Axis forces were heavily censoring their news and reporting for anything that could give any indication of weakness to the other side. Spain was neutral in the war, and they'd suffered heavy losses to the flu, and they were openly reporting it in their newspapers. There were even some editorial comics about it in the Spanish newspapers that I saw in some archives, but I can't find that link anymore, sorry, so I can't show you. I do have some links to some American newspapers, though, which I will have in the sources. In one of them, this army commander is asking a soldier what's wrong with him and calls him a simp. So, it's like the last 107 years just didn't happen. French media picked up the stories and they pointed fingers at the Spaniards, who were naturally outraged, and they pointed fingers right back at the French. Spanish King Alfonso XIII was reported to have a generic flu in September 1918, but this looks like it wasn't like the flu, as in October, it was reported that he had scarlet fever. So it looks more like he had flu-like symptoms for the first 10 days, and then, what he actually had was scarlet fever. Anyways, the name Spanish flu stuck, and it's perpetuated even though it originated in the US and was global by the end of the year. By summer of 1918, the spring wave was in Puerto Rico, the Caribbean, and Mexico. It spread everywhere from Norway to China, India to New Zealand, and then hit the US again within 4 months in the second fall wave. The first wave hit mostly military forts and outposts, whereas the second and third waves also hit civilians, which is when the casualties started to ramp up. Some villages were completely wiped out. New York City lost 33,000 people, and Philadelphia lost 13,000 in a matter of weeks. Some people who died were left in their houses for days, as undertakers and mortuaries were overwhelmed. Burial houses and cemeteries began charging exorbitant fees and making families of victims dig the graves themselves. Canada reported its first civilian victims in the fall of 1918 in Victoriaville, Quebec, where thousands of visitors had arrived for the annual National Eucharistic Congress. Google tells me that this is a big annual Catholicism party where they decide what their public image is going to be for that year. I am not Catholic, so I do not know that lore. Sorry. In all, about 50,000 Canadians died of the flu, which is just under the 60,000 deaths of Canadian troops attributed to World War I itself. It was a mess. There was no coordination between the military and public health officials, and we didn't even have a federal Department of Health until 1919, and it was created in response to the gong show that was the 1918 influenza pandemic.
It's a strange disease with sudden, savage changes.
We all know the symptoms of the flu. You've probably had it at one point or another. I personally have never had the flu, and I know you probably don't believe me, but I never have that I'm aware of. I get other upper respiratory infections at the drop of a hat, like colds and bronchitis a couple times. I've had whooping cough as an adult, but cold-like symptoms coupled with fever, chills, muscle pains, headaches, weakness, and all of that that lasts for a week, never. Influenza viruses are RNA viruses in the family Orthomyxoviridae. Viruses in this family are enveloped, meaning they have a lipid layer outside of their protein capsid. They get this lipid layer from the way they exit your cells after replication. An enveloped virus will typically bud off of your cell rather than burst the cell to escape, like non-enveloped ones do. And in that process, they take a little bit of your cell membrane with them as a coat. You'd think that that envelope might add an extra protection to them, make them a bit more hardy, but it's actually the opposite. An enveloped virus is usually more sensitive to disinfectants, UV rays, dryness, pH, heat, all of that, the things that we use to destroy viruses. They're more sensitive than a non-enveloped one is. Non-enveloped viruses just have their genetic material inside a protein capsule. Some of those that you've heard of like that are rotavirus, aka stomach flu, or poliovirus, which I'm covering in a couple episodes. These viruses can survive on surfaces for weeks to months at a time, while an enveloped one like H1N1 and some human coronaviruses only survive for a matter of days, and that is only in ideal conditions of temperature and moisture. As we know from the SARS-CoV-2 pandemic, transmission from surfaces was not a large factor in the spread of that virus. But there's more than one human coronavirus, and some of them can transmit from a surface. Another non-enveloped virus family that is very resistant to drying and survives very well in the environment is parvovirus, which, if you've ever had a dog, you'll know the danger that that virus represents. The reason why an enveloped virus is more sensitive to destruction than a non-enveloped one is because the disinfectant method, like hand sanitizer, soap and water, dryness, etc., all of those will damage the envelope. And the envelope contains the fusion proteins that the virus uses to enter the cell. Without those, the virus can't infect you. And the lipid envelope is very, very easy to strip away versus the hard protein capsid of a non-enveloped virus, which is more rigid, it will anchor the fusion proteins better, and it's not as subject to drying. All of this to say that both the RNA genome and the envelope mean that the influenza viruses are not that stable in the environment. Remember that. I've already briefly mentioned the symptoms of an influenza infection, but I will recap. You've got your fever, that's 1 to 4 degrees above your normal, which should be around 37, C, so we're talking 38 to 41 degrees Celsius. You've got your chills, headache, fatigue, soreness in the muscles and joints, sore throat, a dry cough, snot and a stuffy nose. You got red eyes and generalized like feeling like a pile of dog shit. It usually takes between 18 hours and 3 days to see symptoms begin after you get infected. And they can last at full strength for up to a week and then end with some lingering effects that can only like taper off after a few weeks. There are some critical complications that can follow for some people at high risk, and normally those people are the very young or the very old who both have weakened immune systems. The big complication is pneumonia, which can be either viral or bacterial, or both at the same time. It would start off as a viral pneumonia and then become bacterial because you've got bacteria all over you, including your lungs, and they get into the tissue damaged from the viral pneumonia, et voila. The bacteria involved is usually Staphylococcus aureus, Streptococcus pneumonia, or Haemophilus influenzae. Remember the name of that last one because I'm going to mention it again in a little bit. Haemophilus influenzae, otherwise known as HIB, you might see it. Symptoms of these two pneumonias are very high fever, dry cough, headache, sore throat, fatigue, shortness of breath, and cyanosis, meaning you turn blue from not getting enough oxygen. With bacterial pneumonia, you can have a productive or wet cough, low oxygen, and high white blood cell counts. A chest x-ray of pneumonia can help distinguish between the two. A viral pneumonia will show a diffuse opacity, sort of cloudiness of the lungs, while a bacterial one will show a bigger clump or plumps of a more solid white, which is the bacterial colonies. Along with pneumonia, some high-risk people like children can develop non-lung symptoms such as heart problems, myositis, which is where your immune system attacks your muscles, which leads to myoglobinuria, or destroyed muscle fibers, myoglobin, in your urine. Sinusitis is infection of the sinuses, and ear infections or otitis media. Like pro tip, if a word ends in itis, it means inflammation, and then the first half of the word tells you what's inflamed. Neurologically, you can develop Ray's syndrome, which is swelling of the liver and brain. You can also develop meningitis or encephalomyelitis. Those two are inflammation of the meninges layer or of the brain and spinal cord itself, respectively. Or lastly, something called Guillain-Barré syndrome. I am going to talk about Guillain-Barré much more in the polio episode. So here I'm just going to sum it up a little bit and say that it mimics polio symptoms with some muscle pain and paralysis or weakness. But this is an autoimmune condition, and the paralysis of Guillain-Barré isn't permanent, whereas polio it is. Globally, almost 10% of the world gets infected with flu every year, and approximately 500,000 deaths will occur, usually of the very young or old, and the immunocompromised. So that is the influenza as we know it today. Let me compare that to the 1918 flu for you. The 1918 flu had all of the same symptoms, but the speed at which it attacked and killed a person was on a dramatically shortened time frame, not to mention the main groups with the highest mortality was different than typical flu. Current flu, like I said, it takes 3 days to appear, a week of full symptoms, and then a couple weeks of feeling poorly, and most people will not die. The 1918 flu did all of that sometimes in 24 to 72 hours before it killed you. The lung hemorrhage was so severe that many victims didn't even have a chance to get to the pneumonia part before their lungs were destroyed by viral replication, and they drowned in their own fluids. 3 days was pretty quick, admittedly. Median time of death was closer to 6 days, but that is still incredibly fast. Hundreds of autopsies were performed during the initial and following waves, and the primary noted finding was destruction of the respiratory tree and death due to respiratory failure. Typical flu mortality is less than 0.1%. That's 1 in 1,000. But the 1918 flu, it was over 2.5%. That's 1 in 40. And the estimated total dead was around 50 million people worldwide. There were under 2 billion people alive back then, so yes, that's 2.5% of the planet that died. The mortality was so high that it lowered the average life expectancy of the US by more than 10 years. What was it, though, that made this flu more deadly than today? The 1918 influenza was an H1N1, and we still have H1N1 circulating today, so what gives? Well, don't worry, because that's what you're listening to my podcast for. I'm going to tell you. So H and N refer to two surface proteins on the outside of the virus envelope. H means hemagglutinin, when you might hear it called HA, and N or Na refers to neuraminidase. As always, clinical names and terms aren't important in this podcast, so I'm just going to call them HA and Na. The surface of a flu cell envelope is covered in tons of these identical HA and Na proteins. HA grabs onto an anchor on the host cell, and for influenza, those are the cells along the surface of your respiratory tract, and it uses that anchor to make the cell engulf the viral particle in a process called endocytosis. All that means is it makes the host cell wrap it in a little bubble wrap layer of the host cell membrane that pushes into the host cell and then floats there like Glinda the Good Witch's bubble. And then the virus escapes from there, it pops the bubble to begin replication. There are other ways that other viruses use to enter, but endocytosis is pretty common. The anchor that influenza uses as a receptor is called sialic acid, and this is very important for determining what species the particular influenza can infect. The Na protein is responsible for cutting the HA away from the sialic acid when it's time to release a new virus particle from the cell. There are 4 main types of influenza, and we give them the letters A, B, C, and D. Influenza A is the big one. It's the one that is categorized by strains of different H's and N's. There's influenza B, which is highly contagious and causes other serious disease, but it only causes small local outbreaks because it does not spread person to person very well. Type C can infect humans and some animals, while D typically only affects pigs and cattle, and it doesn't cause disease in humans that we know of. So far, there have been 18 different HA proteins and 11 different Na proteins as of September last year, 2024, per the CDC. Theoretically, you could have any combination of those 11 and 18, making almost 200 different varieties of flu. In practice, the CDC says there are around 130 different combos that they've identified. Currently in humans, the most common types are H1N1 and H3N2. They are both swine flus, and those each have multiple subtypes and strains. H5N1 and H7N9, both are avian influenzas, have been detected in humans after crossing over to poultry farm workers. Same with H5N6, but again, only in poultry farm workers in China. There's a lot to talk about regarding avian influenza, why it's so deadly, how it doesn't transmit that well, but that's a little beyond the scope of what I'm going to talk about here because our flu, the 1918 flu, was not avian influenza. I will link to a Science Versus podcast episode that's all about avian influenza if you do want some more info on that. I am also not going to bore you to tears by detailing and talking about all the different combinations, all 130 of them, of which H and N's and which species they infect. No. I do want to briefly talk about sialic acid again because that part is important. I mentioned a minute ago that that is the receptor that the HA protein latches onto in order to enter your cells. Sialic acid is a small sugar expressed on the surface of cells in most animals. It's pretty common, and it is attached to other sugars that are attached themselves either to proteins or lipids embedded in the cell surface. So sialic acid bonded to a sugar and then bonded to a protein or lipid. It is the bond between the sialic acid and those other sugars that determine what species an influenza can infect. Avian cells have a straight bond between the sialic acid and the sugars, and human and swine cells have a bond that has a bend in it, like an elbow. So they require 2 different shapes of an HA protein to get in there and grab them. And that's all it takes to change the ability of a virus to infect a species. And now I need to introduce you to another couple of terms, which are the reason why we need to get new flu vaccines every year, and part of why you can still get the flu even if you do get the shot. And those terms are antigenic shift and antigenic drift. I think I've mentioned this before, but an antigen is just something that your immune system reacts to. Usually it's a protein, but it can also be a lipid sometimes. The antigens on the flu virus are mainly the HA and Na proteins, and they can and do change by shifting or drifting. Antigenic shift is a big sudden change, usually in the HA protein that takes it from an H1 to an H3 or an H5, but it can be in the NA too. Antigenic drift is much slower, and it means just small, slight changes within the same HA or NA protein that aren't big enough to give it a brand new number. Shift occurs due to recombination between two different strains of virus, and drift happens due to small random errors introduced during replication. For example, that's how evolution works. This happens quickly enough so that from one year to the next, the circulating viruses are different and that they will escape the immune memory from the previous year's vaccine. Also, vaccines only vaccinate you against the most common circulating strains, not every one that's out there. As I said, there's a few major H and N combination types, but dozens of subtypes and strains. We can't vaccinate you against every single one, unfortunately, unless you want 18 different needles for your flu shot, and that would be very expensive. So they pick the ones that are likely to be the most common the next season. So how does antigenic shift occur? It's actually really fun. You see, swine, pigs, can be infected with both avian and swine influenzas at the exact same time, even within the exact same cell. It could be that pig sialic acid is a bit bendier than human sialic acid, so they can be infected with avian or swine influenza, while birds can only catch bird flu and humans can only catch human or swine flu. Asterisk that a little bit because I'm going to clarify that in a minute. The really fun thing about influenza is that it does not have a single long chain genome. It has a segmented genome. Basically, it's like you. You have chromosomes, right? They're parts of your genome stored on different segments of your DNA inside your nucleus. Humans have 23 pairs of chromosomes, and influenza has 8 genome segments. Sometimes, when a pig has an avian and a swine flu at the same time, one of those flus, when it's packing up to go off and infect another cell, will pick up the wrong segment, coding for an HA or an NA protein, or one of the others. But when it's an HA or an NA, the protein and segments from virus A, and then you have HA protein and whatever from virus B. This is called a reassortment event and can even happen in humans or pigs or any animal that can catch the flu if they catch 2 different flus at the same time. This is generally considered very bad, and when a swine and avian flu do this, this is usually what leads to large-scale epidemics and pandemics.
The awful truth is, I'm not quite all right, and I'm afraid I'm going to ask you to excuse me.
Influenza has been with us since ancient times, and all flus likely originally came from birds and then adapted to infect mammals. Since the year 1500 CE, it has caused more than 14 global pandemics, it being influenza. Since the dawn of the microbial era, which was in 1876 when Robert Koch connected the bacterium anthrax to disease and thus invented medical bacteriology, there have been noted influenza pandemics in 1889, 1918, 1957, 68, 77, and 2009. In fact, the previous influenza pandemics of 1847 to 1851 and 1889 to 1894 very likely contributed to some acquired immunity in people who survived those pandemics, and then may have contributed to a lower mortality rate in people over 35 during the 1918 influenza pandemic. Since we're talking about the origins of influenza, this is a good spot to say that humans gave pigs influenza and created swine flu, not the other way around. There was a guy named Fleming who published a huge list of diseases in pigs in the 1870s, and another named Dimmock who published another list in August of 1918, and none of the diseases mentioned in either book matched the description of influenza in pigs, which is very similar to that in humans. The first reporting of flu symptoms in pigs didn't come until the fall of 1918, so humans gave pigs the flu and named it after them like a bunch of a-holes. The first supposed cause of influenza was identified in 1892 by bacteriologists Richard Pfeiffer and Shibasaburo Kitasato, who were working under Robert Koch in Berlin. They isolated a bacterium that they called Pfeiffer's bacillus, or Bacillus influenzae. And we now know this bacteria by the name Haemophilus influenzae, or H. influenzae, which I mentioned and told you to remember. They were culturing it very reliably from the lungs of people who died of the flu, but they were unable to prove it was the cause of the flu. In fact, what they were doing was culturing one of the causes of the secondary bacterial pneumonia. That is one of the main way we know people died in the 1918 flu. It wasn't the cause of the flu itself, though, as we know now. The actual flu virus itself wouldn't be isolated until the 1930s. In 1918, it wasn't even settled yet that viruses and bacteria were entirely different, or at least the debate was still ongoing, because although viruses had been isolated by filtration starting in the 1890s, scientists of the day couldn't see them. They just knew there was something in the solution that they had filtered that they could cause disease with, but they didn't know what it was. We wouldn't find out what made the 1918 influenza so bad until 2005. And that journey began many years earlier. To get the actual full story of the discovery of the flu's genetic sequence, you're going to have to read the book Flu by Gina Kolata. I will give you the Coles Notes version here because I read this book a long time ago, like a big nerd, and I loved it. It's a good book. But the book itself is well worth the read if you want to learn how medical mysteries and questions like this are solved. But I want that name to stick in your head just for a minute until next episode because I'm going to mention Gina Kolata again when I talk about HIV. The problem with trying to find out what made a disease that happened in the past so deadly is that you need a surviving sample of that disease in order to test. I talked about this during the Black Death, and I will mention it again in a couple episodes. Where do you get surviving non-degraded samples of flu from 1918? Remember, I said it was very unstable in the environment. It does not survive long. We're talking hours to days, right? Well, luckily, the army was very on the ball with taking samples of the soldiers' lungs who had died of the flu, because autopsies were being performed on all of them, and it started on military bases. And at least at first, they were getting the autopsies until the numbers of soldiers dying got to be too overwhelming. They took samples of the lung tissue of these soldiers, preserved them in paraffin wax, and then stored them all in an enormous warehouse. There has always been intense interest in solving the cause of the 1918 influenza. This pandemic traumatized the entire world into constantly fearing it would come back. When it did a couple times, but wasn't quite as bad, people switched from wondering, will it come back again, to, well, it came back and it wasn't as bad, so what's the difference? In the 1950s, Dr. William Hale was visiting the University of Iowa, and during a conversation or discussion of some kind, he suggested that someone should go up north to a community above the Arctic Circle, where the ground was always frozen in permafrost, and see if any samples could be recovered from people who had died of the flu up there. PhD student Johan Hultin got his supervisor to let him put together a group to go and try to find just this, after he was there for this conversation with Dr. Hale. The group involved paleontologist Otto Geist, Hultin's supervisor Dr. Albert McKee, and the pathologist Jack M. Layton. They identified 3 possible sites in Alaska that might be places to start looking for frozen samples: Nome, Wales, and Brevig Mission. They based this on location, as I said, far north where the ground is always frozen, and records indicating high fatalities amongst the Inuit populations. High meaning in Nome, half the population died, in Brevig Mission 90%, and in Wales 55%. In June 1951, an expedition left for Brevig Mission, Alaska. Samples were taken, preserved, and brought back to the University of Iowa, where they were cultured and failed to produce any notable viral illness in ferrets. They thought they had failed in their experiment, and unfortunately nothing else was done in this search for another 45 years. In 1995, Jeffrey Taubenberger, who is an American virologist and current acting director of the National Institute of Allergy and Infectious Diseases at the NIH, was working at the Armed Forces Institute of Pathology, and he started a project trying to recover influenza genetic material from those wax-embedded samples from soldiers that I mentioned. By 1995, PCR had been invented, although it took a hell of a lot longer and was way more expensive than it is today, and Taubenberger was looking for a target to try this new genome sequencing technique out on. By 1997, they had isolated some fragments of the genome from these stored tissue samples, but they hadn't yet been able to get the entire sequence. Taubenberger and his group, including his lab scientist Anne Reed, published a paper on this, and Johann Holten saw it, wrote to Taubenberger, and told him about his trip to Alaska in 1951. Now, while all of this was going on, there was a second group of scientists also trying to find the 1918 flu genetic sequence. And I apologize, I am recalling this from memory because it was closing on 10 years ago when I read Collada's book. I struggled to find a non-paywalled version of an article of this parallel story to refresh my memory with, and I don't want to go back and read the entire book because ain't nobody got time for that right now. But what I remember is this. Alongside Taubenberger's group, which was mostly operating under the radar, this other group was seeking out lots of funding and making a big show in the media of planning a trip to Longyearbyen on Svalbard, an island close to the North Pole, to go and find some genetic material from a person who died up there from the flu in 1918. Same thing as Taubenberger's group, just in Svalbard, not Alaska. If you follow Cecilia from Svalbard on TikTok or YouTube, you'll know that the ground and tundra there is very protected. Like, you cannot drive on it ever, not to mention that in either case, going to Alaska or to Svalbard, you're basically asking people to be allowed to exhume their ancestors. So going about something like this is a delicate process. The group that went to Svalbard took all the latest tech with them. They had isolation suits and clean air tents and breathing apparatus because they were worried that when they opened up the graves, they'd release the flu and people would get sick again. As I mentioned, though, this virus is very unstable and it does not survive to be viable and infectious very long, even when frozen like that. So all of this is going on in the media while Hulton gets in touch with Taubenberger privately and says, yo, I still have a good relationship with the people in Breivik Mission. Do y'all want me to go ask them if I can try again? Taubenberger said, say less, that would be amazing. So Hulton flew up there on his own, got permission quietly from the people living there, found a well-preserved body of a woman who he dubbed Lucy, and took samples of her lungs. He preserved those immediately in fixatives and then flew back down to deliver them to Taubenberger. From there, over the next 8 years, they were slowly able to sequence the entire genome and answer the question of why it was such a horrible disease. The other group that went to Svalbard, they were allowed to excavate some graves, but they were unable to attain any samples of virus. The answer to what made the 1918 flu so deadly has several parts. And forgive me, I'm going to be talking a bit more about, flu virus biology. First, they were able to determine that the 1918 H1N1 was not a reassortment event. It wasn't two flu viruses that recombined together. It had transferred over from birds entirely, just directly, and adapted to humans, likely in that spring wave. Next, as influenza is replicating, the newly assembled virions are sort of lined up along the host cell membrane, attached by the HA protein, right? The HA must then have a chunk of it cut off to release it. The cutting is done by proteases, enzymes that cut proteins in or just outside the host cell. You have a ton of them in all of your cells doing all sorts of jobs. Now, the regular influenza genome doesn't code for its own protease to do this. It relies on your cellular proteases to release it. And the thing about the 1918 influenza is it does this a little differently. It can use either a host protease, specifically one that's embedded in the cell membrane and therefore close and with easy access to the membrane-bound HA, or there is some evidence showing that the NA protein of the 1918 influenza can cleave the HA, which is unusual. All this means a quicker release from the host cell and a shorter viral generation time. The third and final piece to the puzzle is the polymerase gene. This is the replication protein that the virus codes for. All RNA and DNA must be replicated by either an RNA polymerase or a DNA polymerase, respectively. When Taubenberger's group infected human lung cells in cell culture with the 1918 flu, they found that at 16 and 24 hours, viral release was 50 times higher than a non-pandemic control flu. 50 times faster replication and release than contemporary flus that we have around today. Mostly this is due to the speed of the polymerase gene. This is how fast it spread and how it killed within 24 to 72 hours. The reason we don't have H1N1 like this around anymore is because super deadly virulent pathogens like this, they'll always kill themselves out, essentially. From an evolutionary standpoint, it's not good for an infection to be maximally deadly because then you kill all your hosts. So eventually the super virulent ones die with the people they infect, and the less deadly variants or strains that allow people to still be up and walking around and spreading them remain in circulation.
When she wakes, give her some aspirin and cinnamon and milk and keep her here. I'll look at her in the morning.
In terms of how the public handled the flu, well, it was not great, let's say. According to one article in Literary Digest in July 1919, influenza spread was, quote, simple to understand but difficult to control. The cities or towns or states, countries even, whatever level of government, wherever they were able to put in place closures of public areas and impose social distancing measures, those areas were hit less hard by the flu. Duh. In one study that looked at non-pharmaceutical interventions, specifically social distancing, closing of schools, churches and theaters, and public gathering bans, in 43 U.S. cities over September 1918 to February 1919, they found that cities that implemented those measures earlier had a greater delay before reaching their peak mortality, lower peak mortality outright, and a lower overall mortality, i.e., stay the fuck away from people and fewer people die. Gunnison, Colorado, and Fairbanks, Alaska succeeded in isolating their entire towns and reducing their viral burden. American Samoa isolated so hard they didn't have a single case, but a few miles away in Western Samoa, 22% of their population died. Not only did this isolation help with a delay in peak mortality at the city level, but on an individual level. The later a person got sick, both in terms of entire individual waves and then within that wave, the less likely they were to die. So someone who got sick 4 days into an outbreak in a specific area was more likely to develop pneumonia than someone who caught the flu 4 weeks into the same outbreak. The best data on this comes from the U.S. Army. Of the Army's 20 largest forts, in the first 5 of those infected, 20% of soldiers developed pneumonia, and 37% of those pneumonia patients died. In the last 5 forts to be infected, a gap of only 3 weeks, only 7% of influenza victims developed pneumonia, and 18% of those died. That is a huge difference due to the antigenic drift of the virus. One study in Connecticut showed that the closer people were in space to that state's first cases in New London, so the earlier they caught it, the worse they fared. The further away from New London they were, the lower mortality. All this is showing that, as I mentioned above, this was a new virus jumping into humans for the very first time, and as it spent time in humans, it rapidly evolved to be less virulent with every patient and every viral generation. Complicating all of these attempts at implementing public health measures was that the war was still on for most of the first wave, and so leaders and public health officials were very reluctant to put anything into print that could get back to the Axis forces and show weakness. Public health officials were telling people not to worry as the pandemic approached because, quote, fear kills more than the disease. But then the pandemic reaches your city and your neighbors and friends and family start coming down with the horrific flu and dying at up to 40% in some cities, and you start to lose faith in public officials. When there are literally horse-drawn carts moving down city roads with people yelling, "Bring out your dead," I'm not joking, that happened. When that's your reality, but those in charge are telling you not to panic, you're going to panic and you're going to be pissed off. Some people, some children, starved to death because their parents and caregivers were too sick to feed them, and people were too scared to volunteer to help out and feed the children in those situations. This didn't happen in places where authorities were honest and prepared their citizens for what was coming. People and doctors at the time tried everything to cure or treat the flu. But at the time, there were no antiviral drugs, there weren't even antibiotics. And even though that wouldn't have stopped the flu, it might have helped with the bacterial pneumonia. Doctors, they would try bleeding patients, giving oxygen, which wasn't a bad idea. They tried developing vaccines to the bacteria, which they thought was causing it, but that was just causing the pneumonia. People tried treating with salicylates, quinine, codeine, intramuscular injections of silver or platinum, digitalis, aka foxglove, alcamphor oil, or straight up adrenaline, strychnine, arsenic, vapors from aromatic plants, purgatives, sweats, alternating hot and cold showers, iodine, leeches, and antiseptic nasal rinses. One of the only things that seemed to help was blood transfusions from recovered patients, because they wouldn't have known this at the time, but they were transferring the antibody protection. One other terrifying thing about this pandemic that is a little different is that the people who were dying the most were not from the groups you'd expect. If you look at a graph of the distribution of deaths by age group, it forms a W. Normally, these things form a U, with the highest mortality in the very young and the very old. But with this influenza, there was a peak in the middle. Specifically, those under 1 year old in the age group of 25 to 34 and over 74 years of age were hit with the highest mortality. The age group 5 to 14 was the lowest, and 35 to 74 was the second lowest. It's really hard to say concretely why. All we have is our best educated science hunches. From what I've read, the stronger the T cell response to the 1918 flu, the worse the person fared. So most of what we think of as symptoms of the flu or of respiratory viruses and colds is actually caused by our own immune system, not really directly the virus itself. Especially for the flu, as typically the viral particles just bud off of the infected cell rather than bursting the cell to exit that way. Because this virus released 50 times more virus than typical flus, that's a lot more cells being infected, and your T cell response to an infected cell is just to come along and kill it. So this causes massive organ damage and destruction of your lungs by your own T cells. People who didn't have such a strong immune response probably didn't suffer as much lung damage quite so fast. Within reason, of course, because if you have a very weak immune response, like infants or the elderly, then you're not going to do well either. But those people sort of in the middle with an average response, like kids 5 to 14 and people 35 to 74, they had the lowest mortality. People above 35 probably had some leftover protection from the previous pandemics, like I said before. But those 15 to 34-year-olds who were the healthiest and had the most robust immune response, it did not work out so well for them, unfortunately.
If she lasts through the night, she'll live.
Now that I've told you all that, finally I can talk about how the pandemic was portrayed on Downton Abbey. In Season 2, Episode 6, that episode is set in November 1918, and as World War I ends, the family observes a moment of silence at 11 a.m. on November 11th. My bones to pick with this begin now, because we haven't even gotten to the flu. It is November in the UK. It is sunny enough and warm enough outside that people are walking around without coats like it's a pleasant day. Now, Downton Abbey is located in North Yorkshire, somewhere between York and Middlesbrough. In the Met Office weather report that I looked up and saw for November 11th, 1918, the closest to that location I can get for places they listed is Tynemouth in Newcastle. The evening weather there was listed as 53 Fahrenheit, and that's 12 degrees Celsius. And if I'm reading their notation correctly, it was misty with squalls and thunder. So I don't think they were accurate with that weather. There's also been no mention of the flu up until this point, even though by November the first wave was, winding down. The peak of the deadly wave, was September to November 1918. It would have, been wiping out people all over the world, and they probably would have mentioned it. The next episode, though, episode 7, is set in early 1919, and here we have a one-line mention. One. Mrs. Crawley mentions the Spanish flu as being too awful, and that's it. The next episode, Season 2, Episode 8, is when it finally gets to Downton. Most of the episode is taken up with Matthew and Lavinia's wedding plans and with Sybil wanting to elope with Branson, but here we do get some people coming down with the flu. And, okay, you know what? I'm not going to be too critical about how the flu itself is portrayed. Downton Abbey is not a documentary, okay? It's not a show that prides itself on its historical or medical accuracy. It is not The Pit, let's say. It is a romantic historical fiction feel-good soap opera that's meant to be charming and proper and not too graphic. Portraying the flu accurately with how horrific the deaths were and with people starving to death because they were too sick to feed themselves, that kills the vibe. So when the flu does hit Downton, Mr. Carson the butler gets sick, and he's weak and tired, but only coughs once, and then he takes to his bed. Mr. Molesley fills in for Carson at dinner service and ends up starting to feel sick, but it turns out that he was just sampling the wines a little bit too much, and he's just drunk. Several maids start coming down with the flu, and then Lavinia Swire and Mrs. Crawley both get sick. Mrs. Crawley has fever and sweats, but no coughing. Same with Lavinia. They both basically just look really sweaty. Cora, Mrs. Crawley, she takes a turn for the worse and starts bleeding from the nose with a really high fever, and the doctor gives her epinephrine, which is a realistic treatment to have tried, and everything points to Cora being terminally ill. But surprise, it's not her. Cora recovers and Lavinia does not, which is quite devastating and has effects all through the show and next season. This is actually more accurate because Lavinia is the younger person, and we know younger people in their 20s and 30s were the ones to die at higher rates. Mr. Carson and, of course, Mr. Molesley both recover also. Them and Mrs. Crawley perhaps survived an earlier flu epidemic and had lingering antibody protection. So, yeah, it's not quite the vivid portrayal of the flu, but again, it's not meant to be a hyper-realistic show. I will get picky that they couldn't portray the virus correctly in time, and that the November weather wasn't shown correctly for the area, but I'm feeling less critical over how the virus itself was portrayed. It's not really that bad. The timing, it came on quickly and was quite serious. The person who was younger and more healthy didn't make it, but the older people did. Fine. I will give it a 6 out of 10. It could be worse, but it also could be better. And that's all I have for this episode of Tierah Ruins Things with Science. Please remember, I am a researcher, not a doctor, so don't take any of this as medical advice. It is for entertainment only. You can follow me on Facebook, TikTok, Blue Sky, Instagram at Tierah Ruins Things, and you can find all kinds of things at Tierah Ruins Things on Substack, like the show notes and a transcript for every episode, as well as a bunch of other stuff that I post. I share science books and podcasts that I've been listening to. Also on Substack, if you can support me ruining things by becoming a paid subscriber, or you can support me on Patreon if that platform is more your vibe. With either of those subscriptions, you'll get access to upcoming bonus episodes and future Q&A sessions where I answer your submitted questions about infectious diseases. So go sign up. I, myself, do all the research, writing, recording, and editing. The theme song this season was written and provided by iEffigy, and you can follow them on Spotify. There's a link in the show notes. If you have any suggestions for TV shows or movies featuring diseases or pathogens that you'd like me to cover, or questions that you want me to answer for those Q&A sessions, send them in to tierahruinsthings@gmail.com, or just get in touch on any of the social medias that I mentioned. I will see you all next episode, where I will be talking about HIV and the movie Philadelphia.